Can we start, or sure? Is it okay? 1 minute, okay. So welcome everyone after the lunch. I know you had lunch, so typically humans want to go to sleep. I will try to make sure that you don't sleep. So we're talking about stochasticity and randomness. From now on, all this will be again different stories, but all of them will be biological. All of them will be biological. Hopefully, yes. But again, I encourage you, interrupt me, ask me questions, okay? Please. So as you can see, I'm from different background. I'm trying to explain few things, but if you don't understand, feel free to shout and ask what is it, okay? So we will talk about biological things. And we will talk today about signaling profiles in biological development. What is biological development? Biological development, in my opinion, one of the most fascinating phenomenon. It's from how from one or two cells you're creating very complex organisms. Cells divide, eventually they create a very complex organism. This is the most fundamental, in my opinion, the most mysterious biological process, forming of multicellular organism from effectively a single cell. There are many questions associated with the biological development. First of all, how you can create this large-scale organization. And it's typically done by genes, of course, but how? So let me put it in a simpler language, right? So you know what is a gene test, right? Genome test, right? So if I take my piece of hair or skin or nail and do gene test, it will be the same set of genes, you agree? But this is very different from skin, right, or stomach.
So just a moment, we need just a moment.
Nothing works?
I think the live
Technical interruptions. So we're talking about biological development. In other words, question is, so as I said, if I take hair or skin or stomach and do genome analysis will be the same genome, right? Despite the fact that this is different, right? Our hair is very different thing. So the question, how the same set of genes, so the same DNA can produce very different, morphologically different cells here. So that's how the different tissues appear from initially identical, genetically identical cells here. So this biological development is a problem which still captivates people in different areas, from biologists to chemists, physicists, computer scientists, mathematicians. So for example, for physicists, this is a problem of so-called large-scale emerging phenomena. For computer science people, mathematicians, this is the problem how information is passed, right? So it's a distribution of information, because the organism, when it's developed, is essentially information written in DNA. You have to pass it so that you can build properly the organism here. And for engineers, an example of very robust system. We talked about in the first lectures about robustness, remember, right? So these are very robust systems. Okay, good. So let's talk about another important issue which I didn't talk before, universality. Let me try to explain you. So there are many different airplanes in the world exist, right? There are military, civil, small, large, Russian, Indian, whatever, a lot of them. All of them look very different, but they have one thing in common. There is one mechanism how they fly, jet engine mechanism, right? So there is always a question comes, biologists and asking the question, are phenomena in biology the same way? They might look very different, but do they always use the same mechanism or not? So this is a question of universality. So if you ask any biologist, you can ask your biological friends, they will tell you no. Every new biological phenomenon is completely different. If you ask a physicist or chemist who are actually doing biological, they will tell you, yes, there is universality. Okay? I don't know what is the right answer, but I want you to be aware of this universality issue. And fortunately, this biological development is probably universal. Probably we can say with a large degree of confidence, it's universal. And let me show you the picture, which is almost 200 years old. There was a scientist in Russia. His name was Karl Ernst von Baer, so he was a German from a Baltic country, from Estonia. And so these are embryos of different organisms, and you have to look from the top to down, okay? So this will be fish, salamander, turtles, chicken, hog, whatever, right? So you can see there is a clearly similarity, clearly universality, something, right? All these embryos are developing in a similar way. This is, by the way, is actually a very important picture. This is the picture that when Charles Darwin looked at it was one of the things that stimulated him to develop evolution theory. Yeah, so that was Karl Ernst von Baer. This is this person. He was a Russian scientist who made significant contribution to biology. He's essentially a father of embryology, science about embryonic developments. So as I told you that this picture was something, one of 2 or 3 things that stimulated Charles Darwin to develop evolution theory. The most surprising thing that this person was the strongest opponent to Charles Darwin evolution theory. Surprising. So this is a monument to Karl Ernst Baer in Tartu. Tartu is a city in Estonia, which in older time was called Derbt. It has an old university, very nice university. So there is a very interesting tradition. Do you know what is the Walpurgis night? No? Have you Wagner? Do you like Richard Wagner, composer? Do you know? So there is in a German. Okay, I know Indian mythology is extremely complicated. German mythology, they're also complicated, and there's a special night. The special night is between April 30 and May 1st. It's called Walpurgis night. This is the night when all the witches are flying. Anyway, so the students in this university has a tradition, Tartu University. They washing Karl Ernst von Baer with champagne. You see, that's a picture, a real picture. So this monument is really shining because every year on May 1st, they wash this person with champagne. Sometimes with a beer, but mostly with champagne. This is champagne. Good. Morphogens. Okay, let's come back to science. Morphogens. One of the central concepts in biological development is called morphogens. It comes from a Greek words, 2 Greek words, morphos, shapes, gene, development. That was actually proposed by Alan Turing in 1953. So this is a compounds, molecules which are responsible for cellular fate. You can call them dose-dependent gene regulators. So the idea that goes back to Alan Turing was, imagine these are different embryo cells, but all of them identical. Then there is this embryo cells start to produce these chemicals, morphogens, they're diffusing, and then there is a non-uniform potential. And then the idea is if you hear blue one, you will get skin, whatever. That's a general idea development, how it works. He's the most famous person, right? What he did for computer science? No, before. What is the most important contribution to computer science and mathematics he did? Huh? Algorithms. He is the person who essentially started algorithms. But he was a very talented person. He did a lot of things here. He made a lot of contribution to theoretical biology, especially to pattern formation and concept of morphogens. But he's one of the most, not the most important, but the most famous achievement was cracking the code of this machine, Enigma. Do you know what is this machine? Excellent. You're a very educated person. So during the World War II, Germans had a machine which was doing codes, so they could send it to their agents, right? And they thought that nobody could break it. He was able to break it. So the British were able to read codes. And the machine was called Enigma. This is real Enigma, how it looked like. And so it's his most famous achievement, but not the most important from my point of view. So he was really genius. Good. So essentially, starting from Alan Turing and other people, there is this concept of positional information comes in biology. The idea is, as I already mentioned, so these morphogens create non-uniform profile, and this non-uniformity is a way of passing information. This non-uniformity is a way of passing information, and threshold comes into play. Those exactly the same thresholds that I mentioned. So because and if your concentration above this, there is a one gene activated. If between this and another gene, this is called French flag model. That's because the flags are French flag. By the way, this is experimental, you can see. So this is the embryo of fruit fly, and you can see from after some experiment that there some of genes are activated and in position dependent. In this case, this morphogen was called BCD, or bicoid protein, and different concentration of bicoid protein stimulated different genes. Again, thresholds here, there are the thresholds, okay? Yeah, so this idea of positional entropy was introduced by a very famous person called Louis Wolpert. I don't know if in India, he wrote a lot of science popular books. I don't know if they're Indian or not. I have seen them even in Russia. He was very excellent writer, popular books about science. Yeah, so that's Louis Wolpert. Louis Wolpert, and he was born in South Africa, Jewish person, then essentially worked most of his life in the United Kingdom. If you have a chance, strongly recommend you look at scientific books or give it to your kids. They're very nicely written, very pleasant. He's actually also credited with a famous quote. So you can read it here. So it's not, the most important event in human's life is not a birth or marriage or death, but gastrulation. Gastrulation is the process when you are developing different tissues. It's called gastrulation, okay? So those people who are concerned with the marriage, don't worry, the most important things already happened to you. Gastrulation, okay? Anyway.
Professor, you were saying that, what I understood, you said that morphogens decide which genes are turned on and which are off.
They don't decide, but they stimulate a cascade of events that eventually activate different genes. And there is a, yeah, so there is this, it's a proof, experimental proof. You see, so here there's bicoid, and you can see bicoid has different concentration. I can show you a better picture, but you can see this bicoid is more in this end than this end, right? And then eventually you turned, this is one gene, this is another, and this is the third one. It's indirectly,
Did you also mention
fruit flies or worms? That's what they typically think. Fortunately, it's the same events in embryos of humans or chicken here. So he did beautiful measurements. So in this case, this bicoid is a green one. Okay, you can see there is more of this bicoid here than over there. The important thing is that there's a non-uniform concentration, and this non-uniform concentration is a way to pass the information. And there is another proteins called nuclear localization, and there is another on MAPK. Anyway, so that comes to your question. So what people think about this? So one of the most important events, how you can establish this morphogen gradients. You need to have this, by morphogen gradient, I mean non-uniform concentration of the signaling molecules. And the simplest idea, which goes back to Turing, it's something which in this field called synthesis diffusion degradation. This is nothing else as reaction diffusion for some people who already, we talked with some people, right? It's a reaction diffusion. But I also want to notice, don't you think it's the same, almost the same what I told you this morning about catalysis? Remember? Was it diffusion plus death. It's the same thing, right? So the bottom line is, using the same mathematics, you can solve very different problems. Okay, so I write this equation here as a continuous time equation, differential, right? And before I was writing the master equation in a discrete language, but it's the same things. I want you to understand. Okay, good. So essentially the diffusion, degradation, and this is a concentration of morphogens. And if you try to solve this equation, you can get it easily. There will be this decay. Again, it's a stochastic processes, right? Look, the stochastic processes governing these events. And that's what's happening in more microscopically. So there is a set of cells. One of them started randomly produce these morphogens. And then they outside of cells, and they diffuse, and they go to other cells' receptors. They are removed. They removed more. As a result, you will have decreasing the concentration. And then depending on how many of them entered this will be, you can activate one set of biochemical pathway. Here will be another one, which leads to activation of different genes here. Okay, so what's not working? Good. Just a second. Okay, so what are the problems with these fields? So people were able to measure many of these things quite well. And there is a problem. So one of the most studied system is called these bicoid proteins in fruit flies. So when they measure diffusion coefficient, they could measure diffusion coefficients, and they know the total length. Okay, so let's come back to this picture so you will understand. People know the total length of this embryo, okay? They know that bicoid is produced here and eventually has to diffuse. This distance close to 400 micron. So the problem comes is that 375 microns people know. They know diffusion coefficients. Then you can estimate time it takes for the molecules from one end to reach another one, right? You understand, right? And you do the square distance over diffusion coefficient. That's the estimate. But simultaneously, people were able to measure these things here, and it's only 90 minutes. So it's like somebody on a bike moves as fast as somebody on a BMW, which is strange, right? So that's a problem. And this is called paradox. So there is slow diffusion and quite fast formation of morphogen gradients. So this non-uniform profiles form quite fast. People thought about this, proposed several mechanisms, but none of them agrees with experimental observations. Good. So one more piece of information comes, which is given by this very famous person, Francis Crick. So what Crick is famous for? Francis Crick? Good. DNA, correct. So Francis Crick was concerned with the following question. Timing. It takes some time before morphogen gradients can be created. And it is important to have essentially a stationary profile because you need to pass your information robustly, right? You need to have robust information. But how long does it take? It was not clear. Okay, so Francis Crick, very famous person, Nobel Prize for discovery, DNA double structure. By the way, this is a photo that I made myself in a bar in Cambridge, where apparently Crick came, and his collaborator Watson Crick, they came and apparently announced it in a bar. It's a very British way to announce what they did in a bar. So there is a sign here. That's what I made it, that here they did it. Anyway, he was actually a very interesting person. He was very smart, did a lot of things, but he had a lot of strange things. He was extremely critical of religion, especially Christian religions. I don't know why. He also had this a view. He was promoting something that people call positive eugenics. Let me explain it in simple terms. So if husband and wife, both of them smart people, they should have kids. If they're not, they should not. Okay, anyway, this is... But he had Nobel Prize. He could do whatever he wanted at this moment. Anyway, so let's go back to the problem of establishing of the gradient. So essentially, problem comes to the fact how long does it take to establish a stationary state in specific place. So this problem of morphogen gradients, you start everything here, but then eventually your system, how long does it take to establish a stationary state at some location away from a source? And in 2010, very smart work came out, and people were able to establish it. They introduced so-called relaxation function, and they had this time. They were able to, you can calculate time to establish a stationary state in specific location. This is a different time scale. We didn't talk about this. In all the things that I told you, I actually assumed that you immediately go to stationary state. But that's a non-trivial problem. How long does it take to establish a stationary state? And okay, so applying to this reaction diffusion model, or standard diffusion degradation, synthesis diffusion degradation, that's the answers I got it. So this is a time, so imagine this is a distance from a source. This is a time to establish a stationary state, distance x from a source. And so k is a degradation rate, x is a position, lambda is a specific length here. Doesn't look familiar? Remember when I talked about catalysis? Was the same length scale came out, remember? Square root of diffusion over this. Again, I want to emphasize different mathematics. I mean, different problems, the same mathematics. So there is a time scale, so there is a length scale appear. Good, everything is fine. This problem partially explains the problem of fast formation of gradients, but there is something surprising here. It's a linear scaling. So molecules have no bias. They can equally go here or here. So if there is no bias, your diffusion should be square. Size x squared should be proportional to time, not linear. So you expected quadratic scaling. You expected power law 2 here, but you get only 1, which means that there's a kind of a force or potential, and it's not clear where it comes from. The molecules has no bias, these morphogen molecules. Yeah, so this is my naive explanations of, we talked about random walk, but let me try to rewind again. So unbiased diffusion, imagine there is a cat which can go left and right? So then the distance that cat travels in time t scales as r squared. That's because you do a lot of redundancy. You do like this, you do like this. So as a result, if you measure the time, the distance you travel will be r squared will be proportional to t. But if there is some bias, sausage. So then, okay, so probably I better draw a dog because dog has better. But anyway, let's cat who likes sausage, then it will go in this direction. That will be biased diffusion. In this case, distance will be proportional to time because you mostly go in one direction. Okay? So I'm trying to explain what is the problem. The problem was, it looks like experiment shows the morphogen molecules are not biased, but when I try to do the calculations or following these things, it looks like they are biased. So these are the problems. So the measured diffusion constant are too small. Then there is a linear scaling, not clear where it comes from. What is the mechanism of this effective degradation? And we explain this using, again, stochastic model. We can propose a stochastic model, which again is very similar to what was the chemical catalysis I did in the morning. That was the reason for me to give chemical first in the morning and then do very similar, the same mathematics, but different phenomena. There is a model which is exactly burst death with diffusion. So you assume that the signaling molecules produce at the beginning. So here the lattice is semi-infinite. In contrast to what I had in the morning, here the lattice is semi-infinite. Starts from 0, goes to infinity. But otherwise, you're producing these molecules, they can diffuse, and then can be removed, degraded. So it's a reaction diffusion or burst death stochastic process. You can calculate it, again, for this model, you can calculate time it takes to establish a stationary state in location n, and it's again linear scaling. There's explicit formulas, and if you want, you can do a little bit simpler formulas in some limiting cases. In any cases, there is a linear dependence. And before going, there is something else interesting here. Okay, so this is the time to establish a stationary state in location n, and you might ask the question, okay, how this time depends on k, on this degradation rate. And the interesting observation here is that in contrast to naive expectation, if you increase the degradation rate, you actually can reach stationary state faster. So if you increase degradation rate, you can reach stationary state faster. This is strange. Let me give you non-scientific explanation. Was it on TV game here in India? Who wants to be a millionaire, right? Okay. Globalization, everywhere is the same things. So if you have a large degradation rate, then your stationary profile has a low concentration, very low. So it's very easy to reach from 0 to low, it's easy, right? If you need in one day get $100, it's easy, right? I mean, I don't know, 1,000 rupees. You can get from friends, they will give you 1,000 rupees, easy, right? But if you need to collect, let's say, 1 million rupees, it's not easy, right? That's exactly this story. So to go from 0 to something small, you can do it faster. That's explanation. Okay. Good. Okay, so let's come back to this. So this relaxation times are times to establish a stationary state. And so I'm trying to understand why there is this linear scaling comes, right? I said experiments show on the microscopic level molecules of morphogen has no preference. It can go equally probably left and right, whatever. But when I put all the formulas and it's in experimental data, it seems like there is bias. How can I think about this relaxation time? I can think about this relaxation time in the following way. This time, you can write it as 2 terms. So the first term is this first arrival time, mean first passage time. This is related to your question that you lady asked. This is the first passage time. And then And then, so the molecule should come first to this location, then some time for adjustment, and that will be the relaxation time. Okay, and you think about it, so the further away this location, the more important will be this first passage time. So you can do hand-waving argument that this relaxation time can be well approximated by mean first passage time if you're far away from the origin. And it's convenient because you can calculate mean first passage times for this problem. So the mean first passage time to start, if you start here, how long it will take for you to reach the location n. And what we did it, and it's again linear, which is surprising. It's a mean first passage time, unbiased diffusion. It's supposed to be quadratic. How it can be linear? And this slide shows that this mean first passage time is a good approximation. What I plot here, exact analytical calculation for relaxation time versus mean first passage time. And if I go further away from the origin, the ratio goes to 1. So mean first passage time is a very good approximation to So important things, mean first passage time is a good approximation to estimate how long does it take to reach a stationary state, which is an interesting result. Okay, good. So this linear scaling is unexpected, but I was essentially lying to you all the time. The system is not unbiased diffusion because there is a degradation. Degradation does something to the system. Degradation behaves like a potential. It reduces the residence time at each site, and it creates effective concentration gradient. Effective concentration gradient is identical to the force or to potential. Let's try to explain it a little bit more. So imagine that you started at time 0. At time 0, you're producing your morphogens mostly here. As time proceeds, you will have like a kind of a step. Why a step? Because molecules which are a little bit further away will be some of them will be already killed, removed. Okay? And if you continue this way until stationary, there will be a lot of steps, but you don't see the small steps. So you are removing, by degradation removes particle, and the further away you remove more. As a result, you have effective. So each molecule individually has no bias to go, right? But if there is 10 molecules here and nothing here, they will most probably will go there, right? So that's a degradation creates an effective potential. Let me give politically incorrect Texas explanations in other ways. So imagine that, okay, so imagine that I have a gun and I shoot through this door every second, right? And people are running. Only those who really run fast will survive. You understand, right? Or if you're slow, you will be killed. So it's exactly here. This degradation like a shooting. Only those molecules which move faster enough, they will survive. Because the longer you stay at a given site, the more probability you will be removed. So this is another way of looking at acceleration. Degradation works like effective acceleration, like effective potential. And you can actually even calculate this potential analytically in this case, and that's what we did it. Through, if you know the stationary profile, which you know for this problem, logarithm of the stationary profile times kT is the effective potential, and you can do the calculation. This actually means that there is a constant effective force at every location which tries to move particle in one direction. Again, the source is that your system is semi-infinite, and that there is a, if you're moving, the longer you're in a system, the more probability you will be removed, so it creates effective potential in one direction, okay? And that's explanation for linear scaling, right? So remember, in the field with the force, you move faster. It's a driven diffusion versus undriven diffusion, using the language of TASEP. TASEP is driven, right? But it should be symmetric one, will be undriven. So degradation creates these effective driven conditions here. Now, you can actually expand this idea further away, and there are some interesting things. So there are some systems where people are arguing that this degradation is not linear, but power law. So essentially, they're arguing that equation that describes the system, it's again reaction diffusion, but in a slightly different way. There is a power law here. There's the enhanced ligand degradation in a simple molecular way. So the morphogen comes to cell and can only be degraded if another morphogen comes to it, for example. Some other scenarios are possible. Okay.
Is this a power?
Yeah, power. It's a m number. I consider the case so far m equal 1, but it could be 2, 3, 4, whatever. Okay. So this becomes much complicated nonlinear problem. Nonlinear problem, but instead of solving this problem, I can do the same trick as I told you, right? I can look at degradation as effective potential, and I can, because I can, all I need to know is a stationary profile when it will be 0. I can get something like this, and there will be some interesting predictions. So the predictions from these simple calculations are the following ones. So if I have m, this exponent, 0 or 1, then the degradation is very strong potential, and you have a driven system. But if m is larger than 1, it's still unbiased diffusion. But what this potential does, it changes effective diffusion coefficient. It changes the effective diffusion coefficient is faster. Okay, this comes from very simple arguments, and then there was a paper written by mathematicians who actually got the same result through complex analysis. But I got it immediately from very simple arguments. Okay, and this is example of this potential. This is effective potential, the functional location. If it's a linear one, you have very strong potential, but those non-linear are relatively weak. That's why you have different behavior. Okay, so essentially, I would like to emphasize again, we had a stochastic mechanism, right? Very similar to what I introduced in the morning for cooperativity. The same mathematics, almost the same mathematics. I can explain many observations. I can explain how stochastically you can produce this profile, and what is the consequence of this. And well, we argue that these relaxation times essentially are mostly determined by first passage times, and this degradation effectively works as a potential. And we come out with a possible mechanism of acceleration. It's like a potential, so degradation works as a potential, and that's a possible mechanism of acceleration. Again, importantly, stochastic processes are creating complex dynamics in the system. Okay, so the nature didn't care about. The nature doesn't care that if you remove or kill this morphogen, right? The nature wanted to do things to do faster, and to do faster, you do the degradation. There was a problem. You understand all of these things, you are wasting the energy, right? But probably you need to do the things fast. That was the system here. Okay, so this is it. I didn't want to, again, to overwhelm you. So this is a different story with essentially the same mathematics. Okay? The same mathematics. It's a reaction diffusion model, stochastic model, burst death, and you can get, in my opinion, some better understanding. Okay? Any questions, please? I hope you didn't fall asleep. Please.
As far as the morphogen is concerned, it is the production of the morphogen is localized. Is that correct?
Yeah. So people believe that. Yeah. Okay. So this is a good question. So people believe that. So it's some people believe that. Okay. No. Maybe it's even before. Yeah. So people believe that there are several embryo cells, and then at some time something happened in one of them randomly, and they started to produce. And others do not, and that's enough to establish it. That's something what they believe. The story is a more complicated. Biology is a complicated thing, but that randomly you started somewhere and then it diffuses. So it diffuses and removed. You're creating non-uniform profile.
Second thing is in some of the chemical kinetics you had talked about earlier. There were some similarities in the equations with the macro and the micro scale.
Yeah.
And I guess you can go from the micro to macro in some scaling limit. I guess here what I was wondering is the microscopic model you had with the lattice.
Mm-hmm.
Where you have the production queue and then you're hopping.
Yeah.
Yes, this one. Is this kind of a parallel of that?
It's more general. As you understand, discrete phenomena, discrete is always more general. I can always go from discrete to continuous by by making this lattice size smaller and smaller. That will be the limit. Continuous is always approximation. People always forget about. So I don't know about here, but in Russia we taught that it's easier to solve differential equation instead of difference equation, right? But strictly speaking, differential is more general. You can get differential one from a difference, but not another way around.
And one more thing here, the birth is only happening at 0.
That's correct. So it's mimicking what I told you just before, that one of the cells started randomly to produce it, and that's what it is. Right? So, and again, so important things here. This length scale appears, right? This is average distance before morphogen gradient molecule will disappear, and it's exactly the same distance that I had in the morning as communication length in catalytic, right?
So this is the solution to the second order differential equation.
This one is this is actually even easier a little bit. Yeah. So this is you calculate this relaxation function. So this is a stationary concentration. This is instantaneous concentration. You don't even need to calculate. You can do essentially Laplace transformation, and then you can get this time. And then there is from this time this exact formula comes. Right. This exact formula comes. I also derived the formula for discrete version. So this was for continuum version, right? For discrete version, this is the formula that I had. If you want to go to the limit, diffusion much larger than degradation, that will be continuum limit, and you get the same result, right? But there is a Mm-hmm.
Suppose we have a system who has a steady state, okay? To reach that particular steady state, we started with some initial condition, and then with certain MFPT first by time we reach to the steady state. If we change the initial condition, so the transient state, again it will reach to same steady state.
It will have to reach, unless there is some special system bistable, which happens, but not here, you will get to the same stationary state.
So my question is, the MFPT will come same?
Yep. Yeah. It is. It must be the same, yeah. Mm-hmm.
It is the average of all the first passage times.
Yeah. It's average over all possible trajectories.
You are performing multiple simulations, you can perform and take the average.
That's correct.
That's why it comes.
It is called mean.
Right. Mm-hmm.
So mean, what is there?
Mean.
First persistent.
The transient analysis we don't do much in Tesla problem.
It will be transient is bit tough to
There
handle.
is a term for it. The time part.
Questions from this?
It's not chemistry. I hope it's easier for many people, right? But, yeah, please.
Sorry to pester you. Are there any textbook treatments that come to mind that might cover some of these things, quantitative biology here?
I don't know. I can send you my review paper where I try to explain this. But otherwise, not to the best of my knowledge, but probably it is somewhere. So there is a good textbook which is so it's a Conde-Phillips, Conde-Phillips. It's cell physical biology that has a little bit of it, but it's a little bit. Okay, so again, I want to emphasize my point was to present chemical story intentionally in the morning, and to show this exactly the same mathematics, right? With a different biological afternoon. Again, everything is stochastic, and you still can produce things. You can calculate, you can explain the formation of the gradient, you can explain time. Okay?